Good day, students. You are welcome to another video lectures. Today we are going to be discussing about structure and function of DNA and RNA. So we will be discussing under this topic, under these lectures, DNA will make a quick revision on DNA, RNA, and also we are going to discuss about their structure. And also we will talk about the proteins, because these molecules are DNA genetic carriers of genetic information, whereby the whole processes produces protein. And these proteins are very vital for the proper functioning of the body. So DNA, RNA, and proteins are what we call the molecules of life. So let's make a quick review. DNA stands for deoxyribonucleic acid, is the blueprint for life. And DNA molecule is, we already know that it's a molecule of life. And our body is made up of trillions of cells, and each of these cells contain 46 chromosomes. In the humans, so you can see human has 23 pairs of chromosomes inside each cell. And when you open this, the DNA that is contained in the cell is about 2 meters in length. And there are 3 billion DNA subunits, bases, the bases which are A, T, C, G, 3 billion DNA subunits, and approximately 30,000 genes. And these genes are the segments of the DNA that codes for proteins, functional products, which are proteins that perform most of the life processes. So if we look at this diagram, it's trying to tell us that this is a cell, and in the cell we have the nucleus, and each of this nucleus of each cell, there is genetic materials, which is the DNA. And inside this DNA, we have what we call the chromosomes. So, inside each cell, we have what we call the DNA molecules, but we have already known that our cells are made up of 23 pairs of chromosomes. So inside each of the cell, we have the chromosomes, and in the chromosomes, that is where you find the DNA. So when you have this DNA and they have these letters, which are the bases, A, T, C, G, and the transcription, there is a segment of this DNA that is called the gene. And when this gene is transcribed and translated, it gives you a protein which carries out the function of life processes in the cell. So we are going to see those details in subsequent slides. So the DNA structure, DNA is a polynucleotide, in that it's made up of nucleotide subunits, and this nucleotide, each of these nucleotide consists of 3 components. We have the 5-carbon sugar, which is ribose or deoxyribose. In the case of DNA, we have the deoxyribose, and in the case of RNA, we have the ribose sugar and phosphate group. And we also, the third component is the nitrogenous base. So structurally, we have the basic nucleotide structure, which has a nitrogen base, phosphate group, and a 5-carbon sugar. And this is how it looks like. So we have 4 bases that are found in the DNA. We are talking about the and different kinds of nitrogenous bases that are found in the DNA. And this purine, we have what we call purine bases, and we also have what we call the pyrimidine bases. So the 4 bases that are found in DNA, we have the adenine, guanine, which are called the purine bases, and we have the pyrimidine bases, which are the cytosine and thymine. So DNA is complementary in the sense that there is complementary base pairing, where adenine pairs with thymine, and there are 2 hydrogen bonds. A pairs with T, there are 2 hydrogen bonds, and cytosine pairs with guanine via 3 hydrogen bonds. So the stronger hydrogen bond is seen is in the nucleotide pairing CG, because it has 3 hydrogen bonds. So structurally, this is how they appear in the DNA molecule. You have your phosphate group here. These are the phosphate group. You have the ribose or deoxyribose sugar. You have your nitrogenous bases, and A pairs with T via 2 hydrogen bonds, G pair with C via 3 hydrogen bonds, and so on and so forth. And it is when the one strand is going in 5 prime, 3 prime direction, the other strand will go in 3 prime, 5 prime direction, opposite direction. So the DNA structure still discussing, and we know that the DNA is a double helical structure. It has a double helical structure in the sense that it have this nucleotide, and the twisting can be either right-handed twisting or left-handed twisting. So we are going to see all this in details in our subsequent classes. So it was first discovered by Watson and Crick. You remember in our previous classes, we have already discussed that. So let's look at now the replication processes that occur. So when we say DNA replication, the process occurs in the nucleus, because nucleus houses the DNA. So in molecular biology or biochemistry, when we say DNA replication, is the biological process of producing 2 identical copies of the DNA from one original DNA molecule. So it's a biological process that produces 2 copies of DNA that are exactly identical from only one molecule of the DNA. So the parent DNA will now give rise to 2 molecules of the DNA. It's a process that occurs in all living organisms, and it is the essential the most essential part of biological inheritance. This is how organisms get information from their inherit information from their parents. So it's essential during cell division and growth, and also in repair and damage of tissues. And it is a process that ensures each cell to receive identical copy of the DNA. So DNA replication processes occur in stages. In the first stage, we have an enzyme that is called the DNA topoisomerase. DNA topoisomerase is the first enzyme that acts on the DNA molecule during replication. So what does the DNA topoisomerase do to the DNA? It untangles the coils that is in the DNA. So the next enzyme is the enzyme that is called the helicase, and the helicase unzip the DNA molecule, whereby it exposes the strands of the nitrogenous bases for the process of the replication to take place. So in that, when that happens, you have the DNA being separated into two strands. You have what? Two strands that are being produced. So now another then the enzyme DNA polymerase pairs the bases, where it pairs A with T and G with C. And the DNA polymerase also carries out the functions of checking and correcting mistakes that may have occurred during the polymerization, during the addition of the bases. So we have three enzymes. We have the DNA topoisomerase, which untangles the DNA coils, now opening, unwinding the or untangling the coils, because the DNA is super coil. So it's now uncoil it, untangles it. Then the second enzyme is the helicase, which unwind the strand to form two strands. It unwind the double strand to form a two single strand. Now, next one, because the basis has been exposed, so now DNA polymerase, the third enzyme, will now come and act the addition of A to T, G to C, and also it checks where there is mistakes, that is proofreading, check and correct the mistakes. So nucleotides are now paired, and then the fourth enzyme is the DNA ligase, which follows behind and bonds the nucleotides together. So now ligase, now the two strands of the DNA are now created by the ligase. Now DNA replication is a process. The process is fast and accurate, and error occurs in only about 1 of a billion nucleotides. So in about billions of nucleotides that has been added, mistakes or error can occur in one. So, and the process is a kind of very accurate. So replication begins at a specific sites that are called origins of replication. So origin of replication are where replication starts. That's where the replication begins, okay? And the copying of the DNA proceeds outward in both directions, creating what we call replication bubbles. And the parent DNA strands open up as the daughter strand grows on both sides of each bubble. So a eukaryotic DNA molecule has many origins where replication can be initiated at the same time. And this is a kind of advantage whereby it shortens the total time to copy all the DNA. So eventually all the bubbles are merged, and then the end product, which is the two double-stranded DNA molecules, each with one new and one old strand, is being produced. So DNA replication occurs before a cell divides. The process of cell division. So before cell division occurs, DNA replication occurs to have the genetic material being shared. So ensuring that each cells in a multicellular organism all carry the same genetic information. It is also the mechanism for producing DNA copies that offspring inherits from parents during reproduction. The resulting double-stranded DNA molecules are identical. They are exactly the same. So proofreading and error checking mechanism exists during the replication because of that enzyme DNA polymerase. So it has this ability to check and correct errors, what we call proofreading and error checking mechanism. So for RNA structure and synthesis, RNA is ribonucleic acid and uracil replace thymine. Uracil is found in DNA while uracil is found in RNA instead of thymine. It's only in DNA that we'll see thymine. So in place of thymine, in RNA, we are going to see uracil. It's very similar to DNA, repeating subunits and also nucleotides. The differences between RNA and DNA, there are some notable differences between DNA and RNA. First of which is from the ribose sugar. Okay, the 5-carbon sugar that is found in RNA it has what is ribose sugar. So while in DNA it is deoxyribose sugar. Okay, so and in terms of the structure, we can see DNA is double-stranded molecule forming a double helix, and it uses the nitrogenous bases are adenine, guanine, cytosine, and thymine, and base pairing occurs between adenine and thymine, then cytosine and guanine. While in RNA, it's a single-stranded molecule, and it contains ribose sugar, as we said. It uses nitrogenous bases adenine, guanine, cytosine, and uracil. So in terms of pairing, A pairs with adenine pairs with uracil, and cytosine pairs with guanine. In terms of functions, we can see DNA function in storing and transmitting genetic information, and it is primarily found in the nucleus of cells, and replication takes place to ensure genetic continuity during cell division. While in terms of functions, RNA plays various roles in protein synthesis and regulation of gene expression, and it can be found in the nucleus and also in the cytoplasm of the cells. And it has different types such as messenger RNA, transfer RNA, ribosomal RNA, and RNA molecules can be transcribed from DNA. So from DNA, you can produce RNA molecule through a process that we call transcription. So these are all differences, and we are going to see the process of this transcription. In terms of stability now, the differences between DNA and RNA, DNA is more stable than RNA because DNA is double-stranded molecule, and it has the chemical properties of deoxyribose, while RNA is less stable than DNA because it is susceptible to degradation because of heat. So we have already discussed this, and yeah. So now we talk about the central dogma. So the central dogma of molecular biology tends to explain the flow of information, flow of genetic information that starts from DNA, okay, through RNA, and eventually to produce protein. So in all the process, we talk about transcription, we talk about transcription, right? And we are going to talk about translation. So transcription, when you have DNA being converted to RNA. You remember we explained the process of producing DNA from DNA. And when that happens, we can say that is called replication. So DNA to DNA is called replication, while from DNA to produce RNA is transcription. And when DNA is transcribed to produce RNA, when RNA is translated to produce what? Protein. That is how the flow of information, the genetic information in the central dogma molecular biology occurs. So 3 different RNA molecules are involved in protein synthesis. 3 different RNA molecules. We have the messenger RNA molecule. We have which is abbreviated as mRNA. We have the transfer RNA, tRNA, and we have the ribosomal RNA. Okay. These are the 3 different RNA molecules that are involved in protein synthesis, and all of which have different functions, which we are going to be discussing. So in protein synthesis, the first step is transcription, which occurs in the nucleus, okay? And transcription is, we have seen it that DNA is being converted to RNA through the process of transcription. So that occurs in the nucleus. In the second step, we have translation, where the RNA is converted to protein. And this process occurs in the nucleus. So protein synthesis span nucleus, it joins nucleus to the cytoplasm. And we have genetic materials, starting materials in the nucleus. And the end product, which is protein, is now in the cytoplasm. So it means there has to be a kind of transportation of the information, or rather of the molecules, from the nucleus to the cytoplasm. So now the transcription is when DNA is converted to RNA, and it occurs in the nucleus. First, DNA is unwound in a section, and then next step is messenger RNA formed by base pairing. Now, with the parent strand of DNA, it begins the transcription. Now, then messenger RNA carries the message about what type of protein to be produced from the DNA in the nucleus to the ribosomes. We have known the function of ribosomes as organelles. So now transcription occurs once messenger RNA forms. The enzymes in the nucleus remove what we call the introns. And the introns are the non-coding segments of the DNA, and then leave the exons, which are the good ones that codes for functional products rather. So now we have this process explained here. Okay, you have your DNA molecule consists of both the introns, the junks, and everything, exons. Okay, and now transcription occurs. Okay, whereby you have initial transcript. Okay, and then you have what we call messenger RNA being processed, where you have in the poly A tail being attached. Okay, and then when that happens, the introns, the non-coding segment are being removed so that you only have the exon, which are the coding at the end of the matured messenger RNA molecule. So the genetic code now. So how is the information from the messenger RNA now is getting converted to the protein? So we have what we call genetic coding, or rather genetic code. So a genetic code is the instruction that cell uses to translate DNA into RNA molecule. It is made up of codons, which are 3-letter sequence of nucleotides that correspond to a particular amino acid that is going to be found in the protein that is going to be produced. So each 3 consecutive bases on the messenger RNA is a code word that is codon, and it specifies an amino acid. Okay, and the genetic code consists of 64 codons, but only 61 codes for amino acid. While the 3 codes act as either signals to either stop the process, okay, which one coding AUG codes for methionine and is also the start signal for translation. So, and if you look at the characteristic of this genetic code, it is a triplet code. That is, the genetic code is made up of triplets of nucleotides, and it is non-ambiguous. It means each codon specifies a single amino acid. It is degenerate. Also, multiple codons can code for the same amino acid. That is what we mean by degenerate. It is a triplet code, non-ambiguous, degenerate, and also it is non-overlapping, meaning that each ribonucleotide in messenger RNA is part of only one triplet. So, and it's also universal, which means almost every species on Earth uses the same genetic code. So these are the 5 basic characteristics of this genetic code. Number 1 is they are triplet code. 2 is non-ambiguous. 3 is degenerate. 4 is non-overlapping. And 5 is that they are universal. So for the start and the stop codons, the start codon is AUG, and it initiates the process of what? Protein translation, and it codes for methionine. While the stop codons, you remember we said there are 64 codons and only 61 code for amino acid, while the 3 are signals to stop the process or to start the process. For the stopping of the process, it codes AUG are the codes, and it codes for methionine. And methionine is an amino acid that shows the signal that translation to begin. Okay, while the stop codon, when this codon occurs, it means that this translation should be stopped. So whenever messenger RNA is being translated, when you encounter this code, it means the process will be stopped. And this stop codons include UAA, UGA, and UAG. Number 1 is UAA, number 2 is UGA, number 3 is UAG. They terminate the process of protein translation, okay, or synthesis. So this is how the genetic code looks like, okay, and you have your coding and different amino acids that are being produced from that. So in the second step is what we call the translation, where the RNA is being converted to protein. Translation is synthesizing a code from amino acid according to the sequences of the nucleotides in the messenger RNA, and it occurs at the ribosome in the cytoplasm of the cell. And we have what we call ribosomal RNA, and which is needed for protein synthesis. It helps to bind to the ribosomes. And we have what we call transfer RNA, which brings specific amino acid to the ribosome to be assembled as protein. So we have seen the function of messenger RNA, function of ribosomal RNA, and the function of the transfer RNA. So the translation, we have the messenger RNA, and when it enters, we have the small subunit and also the large subunit, and you have your polypeptide being produced. So ribozymes are the sites of what? Protein synthesis. And the transfer RNA molecule looks, it transports molecules that carries specific amino acids to the ribosomes for assembly of the protein that is going to be synthesized or produced. And each of the transfer RNA recognizes the correct codon on the messenger RNA molecule. So structurally, this is how it looks like. So now, the process of translation, you have your free transfer RNA with the specific code that was going to be used to be producing the amino acids, okay? And you have your different amino acids that are being produced from each of these each of these code, and you have your amino acid being synthesized, okay, and the protein gets built up from the different amino acids that are added. So the steps in the translation process summarizes as follow. Number one is messenger RNA leaves the nucleus and migrates to the ribosomes. That's the first step. Then the second step is messenger RNA bind to small ribosomal subunits. Then transfer RNA brings amino acid to the ribosomes, where anticodon on the transfer RNA binds to the codon of the messenger RNA. And the amino acid bonds to it, adjoining amino acid to form a growing polypeptide molecule, similar to the structure that we just see now. And the transfer RNA without the amino acid is released from the ribosome. Then other transfer RNAs bring amino acids to the ribosome to complete the protein molecule. So this is the whole idea behind the protein translation. Okay, you have your messenger RNA here and bounds to the ribosomes. You have your transfer RNA Okay, and you have your transfer RNA adding the molecules of amino acid, and then the growing polypeptide. The growing peptide continues for the synthesis of the whole protein molecule. So the process of the protein synthesis, it occurs in the cytoplasm. Okay, the protein occurs in what? In the cytoplasm. So that is where translation occurs. And you have your ribosomes attached, okay, to the messenger RNA. Now you have your proteins, and then you have your polypeptides. This is a single protein, and you have this is the peptide, and you have many of the peptides that are produced depending on which protein is going to be produced, and you have your polypeptide. So in summary, the protein synthesis occurs as follows, where you have messenger RNA. For the first is DNA molecules being replicated. Okay, now you have DNA being transcribed to RNA, right? You have your DNA producing messenger RNA, okay, in the nucleus. And after that, the messenger RNA is going to be transported, and is going to be bound to the ribosomes. Now, the second step is transfer RNA binds amino acids, and then bring them for the amino acid to be bind to the ribosome. Then you have your growing peptide chain, okay? And then that keeps on going until you have your completed polypeptide chain. So both DNA and RNA have a direction. One end is the 3' and the other end is the 5' end. And then the codons are read in one direction only. There is redundancy in the genetic code. It means different sequences can specify for the same amino acids. Now, for the mutation, when there are changes in the nucleotide sequence of the DNA, we said mutation occurs, and it can be passed from one generation to the next generation, to future generation. So we have different types of mutation. We have the single base substitution, which we call point mutation. Is a single base substitution. For example, when we said, assuming this is our genetic code, in the sentence that reads, the cat saw the rat. Okay, the cat saw the rat. And now there is a single substitution of the nucleotide. Instead of R here, it is replaced with H. It is substituted with H. So now the sentence will read, the cat saw the hat. So it means the entire sentence changes the meaning. That's how mutation changes the information that is carried in the DNA. Okay? So you see how deleterious mutation can be. So there is also deletion, where a small DNA segment is lost. There is also an insertion, where a segment of DNA is added. There is also what we call frame shift mutation, which is modification of the reading frame after a deletion or insertion, resulting in all codons downstream being different. There are also mutations that are called silent mutation. Okay, silent in the sense that a change in that nucleotide sequence did not necessarily bring a change in the resulting amino acid. So that is a silent mutation. So we have what we call somatic mutation. It occurs in the body cells or cells that do not lead to the production of gametes. So somatic mutation that occurs in leaf, roots, or stems are usually not passed on to the future generation. So that's all. That's where we are going to stop. I hope you really enjoyed this lecture. If you are yet to subscribe to the channel, kindly subscribe and put on the notification bell so that our future content can be, you will be notified when we upload our future content. Thank you for watching and see you next. Bye-bye.